Good morning, everyone. I am Frédéric Rioloca. I am a research director at the Imagine Institute. And today I was asked to talk about the DOCK11 deficiency, a new X-linked actinopathy with immune dysregulation that we described with Charlotte Boussard, a pediatrician, doing her PhD in the lab. This work was performed at the Imagine Institute, and on the first slide you can see the team members that are both clinicians, pediatricians, and research scientists, and sometimes doing both activities, like Charlotte, who is a pediatrician doing a PhD of science. So today we are going to discuss the immune-related actinopathies that are primary immune deficiencies and dysregulations, so PIDs. There are 23 monogenic causes that were known so far before the description of DOCK11. And thanks to the next generation sequencing, there was a burst of genetic discoveries in the last 10 or 14 years. These mutations have various impact on cytoskeleton functions that can be actin itself or actin elongation, branching, capping, but also the regulations through raw GTPases activities. Mutations on the genes that are depicted here on the slide are leading to various clinical consequences that could be severe combined immune deficiencies in the group 1, that is basically CORO1A mutations, but it could be also combined immune deficiencies like for the DOCK2 and DOCK8 mutations, and it could be also antibody deficiencies, immune dysregulations, phagocyte defects, and autoinflammatory disorders. We will first start with the first description of immune-related actinopathy. That was mutations in the WAS gene, described by the end of the 80s, beginning of the 70s. And in fact, these mutations are now better understood and they are more complex. So the loss of functions of mutations of WASP, so that are depicted in red in this cartoon, are leading to a combined immune deficiency, so T cells and B cells deficiencies, together with thrombocytopenia and small platelets, and autoimmune manifestation. Hypomorphic mutations related to the type of mutations and sometimes with somatic reversions are leading to X-linked thrombocytopenia, so there is a partial correction of the defect, mostly in the T cells and B cells, but the platelets remains affected. And then the last type of mutation is the gain of function mutations that are depicted in green on this cartoon, and that are affecting uniquely the JBD domain. And these mutations are leading to X-linked neutropenia, meaning that mostly the neutrophils are the cell types that are affected with these mutations. Now we are moving to the DOCK-related cytoskeleton functions. So the DOCK family is a group of proteins that are guanine exchange factors, grouped by subfamilies in the depending of their domains. The DOCK-A subfamily that contains, in addition of the DHR1 and DHR2 that are common to all DOCK families, they are having a SH3 domain and a helical domain, as well as a PBR domain that is defining these subgroups, in which we find the DOCK2 protein. In the DOCK-B, that will not be the topic today, there are DOCK3 and DOCK4. In the DOCK-C subfamily, we can find DOCK6, DOCK7, and DOCK8. And as I will show you, there are known mutations of DOCK8 leading to immune deficiency and immune dysregulation. Finally, the D subgroup is composed of DOCK9, DOCK10, and DOCK11. And so far, I mean, until the last 3 years, there was no mutation known to affect these proteins in human diseases. So as I said, these proteins are guanine exchange factors, so they allow the transfer of GTP instead of GDP to provide the activation of many small G proteins that are depicted on the left on the slide. They are involved in cytoskeletal functions, as it was here summarized in this review. And we can see that the WAS protein is interacting with the DOCK8 and DOCK2, and that all these proteins are very important for the shaping, the morphology, the motility, and the cellular functions. The first one is the DOCK2 deficiency, so that was described in 2015. It's an autosomal recessive actinopathy with invasive infections. So as it is shown here in this paper, so there were recessive mutations in consanguineous pedigrees or in non-consanguineous pedigrees with compound heterozygous mutations that were affecting different domains of the protein and that were leading mostly to the absence of the very low expression of DOCK2 at the protein level. The consequences were a reduced RAC1 activation as well as a reduced chemotaxis of T cells and B cells. So therefore these lymphocytes were less able to reach the peripheral tissues, explaining some of the clinical phenotype observed and in particular the infections observed in these patients. There is also an impact on the polymerization, the actin polymerization, of T cells and B cells. And as depicted on the right panel, it is shown here how DOCK2 is interacting with the WAS and how it has a role in CD19 and CD21 expression in B cells that are therefore reduced when DOCK2 is absent, and that is leading also to a marginal zone B cell deficiency. So overall there is a deep humoral response deficiency as well as a T cell deficiency, so explaining the infections observed in these patients. For the DOCK8 deficiency, so it's also an autosomal recessive actinopathy with combined immune deficiency, hyper IgE, and severe atopic dermatitis. So there were also homozygous or compound heterozygous mutations leading to a complete or partial defect of DOCK8. What is interesting is to observe that it was affecting the T cell development. As you can see here, there is a reduced T cell counts in the patient that were studied in this initial paper in 2009. And a very marked feature of this patient that they have hyper IgE and hyper eosinophilia, and that is explaining the severe atopic dermatitis as shown in these pictures in patients with DOCK8 mutations and a defect in skin antiviral immunity. Similar to the WAS genetics, there are somatic reversions that were discovered in the DOCK8 deficient patients. So as shown on the left of the cartoon, we have a somatic reversion that was mostly observed in the T cells of the patients and not in the B cells of the patients, and but that may vary from patient to patient, and that is leading to a partial or important correction of the genetic defect of the DOCK8 deficiency, leading to the reduction of allergy, the reduction of atopic dermatitis. There was also less viral, bacterial, or fungal infection, as normally it is observed in DOCK deficient patient, and they have an increased antibody response and therefore an increased survival. So now we are moving to the topic of the day, DOCK11, that is also a GEF protein involved in immune function. So that was described in cellular models, and there was also a DOCK11 knockout mouse model, but without a particular phenotype, an immune phenotype. Only a reduction of the B cell compartment, but without a clear impact. So DOCK11 was in this review described to be associated with DOCK8 through the activation of CDC42. So very few was known when we discovered the first mutation of DOCK11. So the DOCK11 is on the X chromosome, so therefore it's a new X-linked actinopathy with autoimmunity as observed in these patients with severe ITP, autoimmune hemolytic anemia, Evans syndrome, but also autoimmune neutropenia and autoimmunity against the gut. So these mutations were affecting the different domains of DOCK11, the DHR1, the DHR2, or they were making the structure less stable. There was a minimal impact in the described mutations at the level of protein expression, sometimes a little bit reduced, but not always. DOCK11 deficiency clinical and biological features were depicted on this table. We first can see that they all presented with autoimmune cytopenia related with autoantibodies, sorry. But these patients presented as well skin manifestation that could be very severe, sweat syndrome, folliculitis, bullous and cutaneous disease, digestive manifestations with in patient A, a very severe IBD. And then regarding the therapies of these patients, we could see that they were mostly treated and not always efficiently with immune suppressors, steroids, rituximab, and other specific or targeted therapies, but that were not that efficient, and we will come back to that topic at the end of the presentation. On the immunophenotyping, the most notable observation was the excess of B cells that are CD121 low, so they are supposed to be enriched in autoreactive lymphocytes. And the immunoglobulins were normal or slightly decreased, and again there were a lot of autoantibodies in these patients. The impact on the morphology of the platelets was shown by electron microscopy with more elongated platelets and more activated platelets. Showing that the DOCK11 mutations were clearly impacting the morphology of the platelets, and not always their function, as not all the patients had, so they could have thrombopathy but without bleeding. The DOCK11 deficiency consequences of the morphology of BVB cells are depicted here. So you can see on the left panel the control cells, and on the right panel the patient cells so we were analyzing the spreading, and as you can see in the middle of the panel, these very hairy BVB cells with there was an increased protrusions and abnormal filopodia-like structures, and we observed also an imbalance Rho-RAC activity. The DOCK11 mutations on the lymphocyte motility were tested on this experiment of migration, and in fact when the cells are moving, so there is an important actin cytoskeleton remodeling, with ROA activity at the rear of the cells for the retraction, and a strong RAC1 CDC42 activity on the front of the cells in order to make the cells moving. So when we measured the migration speed, so in all patients we observed an increased motility of the lymphocytes of the patients, and that can be mimicked with a RAC inhibitor. We could make a phenocopy by using this drug in vitro, so showing that when we reduce the RAC1 CDC42 activity, there is an increased speed of the cell. And importantly, we can see here the actin polymerization and distribution of the cellular in the cells. And we could see that whereas in the healthy control there was a regular distribution of the actin depicted by yellow and red colors, this actin was mostly at the rear of the cells in the patient cells. So there is an increased migration speed and an abnormal actin distribution as a consequence of the DOCK11 mutations, and we could mimic that by doing a knockdown of DOCK11 in primary cells. In the patient cells, what we observed as an important phenotype is a reduction of the FOXP3 expression in Tregs, as shown here on the right panels, where we looked at the FOXP3 and Helios expression, that are the two master transcription factors, in the Tregs, and as you can see on average and in the middle, there is a reduction of this FOXP3 expression. And that was related to a decreased phosphorylation of STAT5 in response to IL-2, and as you can see, the conventional T cells were less proliferating in response to IL-2, as compared to the control. That was true for the T cells, for the activated T cells, but also for the mate cells, and in a less important way to the B cells. So these DOCK11 deficiencies were leading to a very severe phenotype in some patients, and as shown here in the patient A, this patient suffered for a very severe autoimmune IBD and autoimmune and inflammatory bowel disease, and that led to the recommendation of bone marrow transplantation, that he was transplanted with an unrelated donor, but that was fully matched. So there was the correction of the cell morphology and the Tregs in the patients after bone marrow transplantation, as seen in these stainings, but there was a digestive relapse after a year of honeymoon, so that is raising the question of whether DOCK11 may have a non-hematopoietic function. And as you can see here, DOCK11 is indeed expressed in many other tissues, and in particular in the gastrointestinal tract, so meaning that the transplantation of hematopoietic stem cells might be not sufficient to cure the DOCK11 deficiency. So on the conclusions of this DOCK11 actinopathy, it's a new X-linked autoimmune actinopathy that we found in 8 patients. It was early autoimmune manifestation with cutaneous, digestive, and manifestation and functional thrombopathology. DOCK11 deficiency leads to the dysregulation of the Rho GTPases balance with the Rho RAC CDC42 balance. Cell morphology and motility are impacted as a consequence of the actin cytoskeleton remodeling defect, and mostly associated with the CDC42 deficiency activation. There are abnormal regulatory T cells and a reduced IL-2 STAT5B activation. So therefore, for the therapeutics of these patients, there is the question of the bone marrow transplants or maybe targeted therapies, but more work has to be performed in order to better understand the pathophysiological consequences of the DOCK11 deficiency. And to conclude, there is also variable clinical expression in the DOCK11 deficiencies. The group of Can Bostook has described patients with DOCK11 hemizygous mutations leading to a mostly complete deficiency of the DOCK11 expression at the protein level. And these patients were developing systemic inflammation and normocytic anemia, a phenotype that was quite different from the patient we described in our paper. And there is a very nice article by Stuart Tangier and Isabelle Metz making the description of these two deficiencies and pointing to the fact that probably the consequences of the mutations are related to the different phenotype with normal expression, missense mutation with normal expression are leading to autoimmunity, at least in the group of patients we have described, whereas the complete defect is rather associated with a severe inflammation and anemia. There are no new mutations that are found by exome sequencing, clinical genome or whole genome sequencing. And the question is how to validate these mutations. So far, all the mutations were leading to a reduction in CDC42 activation, but it's an assay that is not easy to perform. And we found these different mutations in Evans syndrome, in a syndrome with autoinflammation, inflammatory bowel disease, deformative polyarthritis, and meningoencephalitis, showing again a very broad clinical spectrum in these mutations that have to be validated. So there are still a lot of questions in these diseases, and more work has to be performed in order to fully understand the implications of this DOCK11 deficiency in the cellular functions and in particular in the immune cells. I thank you very much for your attention.